Alrighty, hi guys. Welcome to my video DNA replication part 1 in the cell. So this video is going to cover how DNA replication happens inside a cell. If you want to learn how it happens outside a cell, namely PCR, you can watch my second video in this series, DNA replication part 2, PCR and amplified products. Alrighty, so in this one we're going to talk about the process of how DNA replication happens inside the cell, as well as what enzymes are involved in it. And I'm going to try and get into the details of things that people usually don't understand very well, or the readings don't usually cover, and people usually get the wrong ideas. So I'm going to make sure to hit some of those things too. So let's see how this goes. Alrighty. So as you recall, so in week 2, we talked about how, DNA is a double strand. It's got these bases across the middle here. Oh, that's beautiful. This is why I'm not an art major. All right, here we go. Yeah, DNA ladder. Okay, so there's our DNA strand. Right, so it's double stranded here. And we also know that a chromosome is a big long strand of DNA, right? And that prior to meiosis and mitosis, which we learned last week, you have to duplicate the chromosomes. You have to duplicate the DNA so that when you finish cell division, both cells have a full set of the original DNA. So we have to make an exact photocopy of the DNA in one cell so that we can give it to the next cell. And this week, we're going to talk about how you do that. DNA is designed in a fantastic way. God is a genius because it makes this process really, really easy. The cool thing about it is, so DNA is double-stranded. And as you learned, it's got these bases across the middle. And there's the base pairing rules, right? So if you have an A on one strand, you have to have a T on the other one. If you've got a C on one strand, you have to have a G on the other one. So, so by having it in this setup, you only need God made it so that you only need to know the sequence of one strand in order to figure out what the sequence of the other strand is. Meaning that if I separate these two strands, I can read one of them and build the other one exactly like its twin, or exactly how its twin was, without ever having seen the twin. Now, sorry, that may have been confusing, but hopefully it'll make more sense here in a sec. So the idea behind this is, picture the DNA like a zipper, right? Like on a standard jacket. The idea here is we want to unzip this thing so that we can actually get to the bases that are hidden in the middle here. And then we are going to read each of the two strands independent of each other and build a new strand complementary to it. Let me show you how we do that. In order to make any reaction in the cell happen at a reasonable rate, especially something as complicated as DNA replication, we have to use some special proteins called enzymes. An enzyme is simply a protein that makes a reaction happen faster. Makes a reaction happen faster. Okay, because most of the biological most of the reactions that we use in biology, the chemical reactions, would take an eternity to happen on their own. Like building a DNA strand. Imagine trying to have a DNA strand just spontaneously make itself. That would take upwards of 1 million years to make one base stick onto the strand on its own without help. So the goal of the enzymes is to be what we call a catalyst, which is basically make it happen faster than it would normally. So its job is to just speed things up. So the reaction would happen eventually, hypothetically, but the enzymes are trying to make things speed up. So like with DNA replication, one of the enzymes we'll teach you about here, the enzyme responsible for adding DNA bases on, it would take, yeah, hundreds of millions of years for a DNA base to randomly add itself on. But this enzyme that I'll teach you about, DNA Polymerase III, can do 1,000 bases per second. So they really speed these reactions up, and that's why we rely on them so much. And each enzyme is extremely specific to a specific reaction. So each enzyme only knows how to do one thing. It does one reaction and that's it, which means we have to have a different enzyme type for every single chemical reaction we want in the body. That's a lot, and that's a lot of enzymes. And that's why we have so many genes. Not all proteins are enzymes. There are quite a few proteins that are not enzymes, but our body has or requires quite a few enzymes in order to function. We're only going to learn about a few of them in this class. But because there are so many steps to DNA replication, there are a lot of enzymes that are involved, and we will expect you to know each of the enzymes and what they do. So I'll make sure to go over those as we go here, okay? All righty, so let's pop over to my crude little simulator here. I'm still working on it, so bear with me. But hopefully it will help you get the idea of how this process works, and we're gonna work through this on a step-by-step basis. Okay, so just to orient you here, this stripe here across the middle, this is representing our double-stranded piece of DNA, and you can imagine the bases across the middle here, okay? So here, the dark blue one's the top strand, and the light blue one's the bottom strand. Notice there's my 5 and 3, 5 and 3. Okay. So the first step, the first thing we have to do in DNA replication in the cell is to unwind or unzip this DNA strand so that we can actually read the bases in the middle. Now, unlike a zipper on a jacket where the zipper starts at the top and goes to the bottom, meaning we would expect a zipper to come over here and go all the way over there, start at one end of the DNA strand and go to the other. This actually is going to work more like a backpack zipper, where the zipper is actually going to start in the middle, and we're going to put two zippers there, and they're going to go in opposite directions. And I'll show you why in a little bit. Okay, so in the middle of this strand here, at various points throughout the chromosome, let me pull up my drawing tools here. Okay. So at various points throughout a chromosome that's going to be replicated, we have these spots called the origin of replication. Very creative. Or sometimes, especially when we get into the more complicated stuff later on in the semester, you'll see this abbreviated as ORI, or sometimes ORIC, but usually just ORI. So that's the origin of replication. And that's simply, where do the zippers start? That's all it is. It's where do I start unwinding this strand? That's what the origin of replication is. Now, so specifically, the zipper, so I keep talking about this zipper. There's an enzyme that serves as our zipper because we need to unwind, we need something to unwind this DNA, but it's not going to do it on its own. So we need an enzyme to serve as the zipper or the unzipper. And the name of that enzyme is called Helicase. Oh no. Hold on, give me a sec. Okay, I think I fixed it. All right, so we have an enzyme called, we have an enzyme called Helicase that's going to hop on here. So pretend each of these rectangles is Helicase. They represent Helicase. What Helicase really looks like is it's really like a donut. So if I were to draw this, and let's see here, let's stick with our colors here. So if this is like my double helix, and I think so, well, Linking that out there. Okay, here's my little very crude DNA helix. Okay. Helicase is actually like a little donut enzyme that comes on here, and it's going to grip, it's going to wrap around the DNA strand. So the DNA strand, or the DNA double helix, is going to go through the middle, or it's going to go through the donut hole like that. So this thing wraps around the DNA strand, but given that this is in cross section, you can image that or imagine that. So this is the helicase right here. So at the origin of replication is another word for the helicase binding site. That's where helicase is going to stick to the DNA. Now notice here that the helicase, I have two of them, right? It's not just one. There are actually two of them. Now, in a lot of the videos and the pictures that we show you, they will show it something like, they will show you a picture something like this, where this blue donut represents my helicase, and it makes it look like there's only one. But that, I find, confuses people. It's a simplification because we've got the same thing going in both ways. But I find that it makes more sense to people to show them what's really going on. So let me show you what's really going on. There are actually two helicases that hop on at the origin of replication back to back with each other, just like you have two zippers on a backpack. And one of the helicases is going to start at the origin of replication and go that way and start unzipping that way. And the other one's going to start unzipping that way, just like a backpack zipper. So let's let it do that. So helicase is going to come in here and it's going to start unzipping the DNA. I'll come back to that one here in a sec. Okay, here we are. Okay, so helicase is going to start unzipping the DNA. So it's going to form this little thing here. Notice that we've got our double-stranded DNA coming in here, and then we've separated the two strands. So I've got one strand up here, and I've got the other strand down here. They're not touching each other anymore. I broke all the hydrogen bonds between the bases. They are now separated from each other. And these helicases are going to keep going until they run into another helicase or they fall off the back end. So they're just going to keep right on driving until we're unzipped. But for the purposes of my little simulator, pretend they're going to keep going on forever and ever, kind of thing. Okay. So we've unzipped this. Now notice we've formed this little shape here with the DNA. We've formed two of them. One right here where we've got like a little split over here. And we've got a little split over here. These are called replication forks. And there's always, the replication fork is always going to be right behind the helicase because it's wherever the DNA is currently being split. So there's two replication forks for each origin of replication. We've got one following that helicase and one following that helicase over there. So these are the replication forks there and there. And we call them that because it's kind of like a fork in the road, right? If you were driving here and then you had to choose whether to go that way or that way, it's like a fork in the road. So we call it a replication fork. Okay, so yeah, the helicases are going to be unzipping. Now let's come back and explain this guy. His name is really long, so it's abbreviated here Topo because I couldn't fit it. His name is Topoisomerase. That's a really long name. But his job is kind of obscure, but it's very, very important. And let me show you a picture here to help me make the point with an analogy. Okay, if you've ever taken, so this is the analogy here, but it helps you understand why topoisomerase is important. So if you've ever taken a rubber band, here's a little experiment for you to try. If you take two rubber band pieces and you wrap them around each other like you see up here, so they're twisted around, they're coiled around each other. And then you have somebody hold one end together, and this works with ropes or anything. It's really interesting to try it out. So you coil two of them around each other, and you have somebody hold one end of the coil, and you have the other person take the other end and try to just pull them straight apart. So don't try to unwind it, just pull them apart, and see what happens. What you'll find is, so you'll pull this end apart over here, but it's going to get harder and harder. You won't be able to pull it all the way back to the other person's hands. You're going to get stuck because ahead of the fork here, you're going to get what we call supercoiling. It's going to get tighter in front of the fork over here. So the cords will actually wind tighter around each other, and you'll start getting these little kinks here in the strand, and eventually you won't be able to pull it apart any further and you'll get stuck. DNA does the exact same thing. So if we've got helicase over here at the fork, here's my little helicase right here, and it's going this way, and it's trying to unwind the DNA strands, the DNA in front of it is actually supercoiling. And so helicase won't be able to make it very far without getting stuck. So the job of topoisomerase is, if I've got a DNA strand up here, so he travels in front of DNA helicase, so there's my topoisomerase. His job is to travel right in front of the helicase and fix the supercoiling so that helicase never gets stuck. He actually does that by, so if we've got my DNA strand here, he actually comes in and he makes a little tiny cut in the DNA strand, or in one of the DNA strands, and lets it spin around to get out the coils, and then he fixes it again, and then he travels a ways down and he makes another cut and lets the spins come out and then puts them back in again. So he's constantly, so he's traveling ahead of helicase. So while helicase is unwinding the DNA strands, he's taking the kinks out of the DNA strand in front of helicase, so helicase doesn't get stuck. That's the job of topoisomerase, is to travel ahead of helicase and make sure he doesn't get stuck. A big long name for a small guy. Okay, let's pop back over to our simulation here. So yeah, so there's a topoisomerase in front of that helicase, making sure he doesn't get stuck, and there's a topoisomerase in front of that one to make sure he doesn't get stuck. And we form the replication fork. So topoisomerase and helicase form the replication fork. Now we have a problem, because DNA does not like to be single-stranded. It really, really doesn't. It has to do with the nature of the DNA bases, the nucleotides. They don't like water, and your cells are full of water. So they are designed, a DNA strand is designed to hide the bases in the middle where the water can't get to them. So the bases are all hiding in the middle where the water can't get to them. And then you put the backbones that are okay with water on the outside here so that the water interacts with them, but they keep these guys safe. So what happens when you unzip these two is these bases are extremely uncomfortable because now they're exposed to water. They're not being hidden anymore. I pulled them out. There are bases poking out the bottom here. And so these bases desperately want to be double-stranded again, and these bases desperately want to be double-stranded again. So what's the natural thing to do? Stick back together again. So essentially what we've got is, imagine a backpack zipper where the zipper sides are magnetized to each other. You would unzip the backpack and it would re-zip itself behind the zipper. We don't want that to happen. We want these strands to stay separate from each other long enough for me to actually duplicate the DNA. So we have to have something to come in here to prevent them from remagnetizing to each other behind the helicase after we've unzipped it. Otherwise, we're not going to get anywhere. So let's introduce our next one. Now, this one isn't an enzyme because it's not going to make a reaction happen. This one is called single-stranded binding protein. We're extremely creative with this one. Single-stranded binding protein. Also abbreviated SSB. And because his name is so long, you'll usually see SSB or SSB protein. But that means single-stranded binding protein. So here's our little SSBs. Notice there's a bunch of them. And their whole job is to go grab onto these single-stranded pieces of DNA, and so binds to them, hence his name. He binds to single strands of DNA. And his whole job is to grab onto them and make sure he doesn't stick to the other guy. That's his whole job in life. He just grabs onto the single-stranded pieces of DNA and makes sure he doesn't go stick to the other strand of DNA again. Make sure he doesn't glue back together again. That's his whole job in life, is to keep the strands separate once we've unzipped them. So single-stranded binding protein, you get a bunch of these. Notice there's more than one. You get a bunch of these that just come and just coat the DNA molecule to keep those bases happy so that they won't go and stick to the other strand again until we're ready for them. So that's what the single-stranded binding protein is for. Okay, so now, now that we've formed the replication fork, we've separated these things. So these three guys, their whole job is to help me form the replication fork. These guys actually peel it apart, and these guys make sure it stays peeled apart. Now we have to actually start building a new strand, because the whole goal is to take one double helix and turn it into two double helixes that are identical to each other. So what we're going to end up doing is reading this top strand and making a second strand to go with it, and reading this bottom strand and making a second strand to go with it. And in the end, we're going to wind up with two double helixes. But so we're going to have to build a new strand down here. And the guy that's going to do that, his name is DNA Polymerase III. I know, that's a crazy name, isn't it? DNA Polymerase III. His job is to build the DNA strand, the new one. We call it the complementary strand. So his job is to build the new strand. But he's got this one little caveat here. He doesn't know how to start a new strand. He only knows how to make a strand longer. Isn't that weird? For some reason, the way God designed him, he doesn't know how to start a new strand. He only knows how to make a pre-existing new strand longer. So we've got a predicament. We need this guy to build me a new strand right here and down here, but he can't start a strand, and I don't have a strand started. So somebody else has to come in and step in and do his job, and get the strand started so that he can finish it. And that guy is an enzyme called Primase. And his job is to build a short little piece of the new strand called a primer. So there's a tricky thing. A primer is a nucleic acid strand. Primase is an enzyme that builds a primer. Okay, so the primase enzyme builds the primer. And the reason we call it a primer is it starts the strand. If you've ever seen old-fashioned water pumps from like the Wild West stories, they've still got them in some places nowadays, where it's got like its little handle coming off the top here, and a little spout coming out the side, like that. Oh, that's actually pretty good. Not bad. Oh, now it looks like a weird bird. Okay, anyway. So then the goal is you pump this handle and you make water come out this part right here, right? Water. Woo. But if you've ever seen, there's an old church film called The Pump. And one of the parts to it points out that in order to make these old-fashioned pumps work, you had to actually add a little bit of water to it first. You had to pour some water down inside it first in order to get it to pump more water out. Otherwise, it wouldn't work. And the amount, and when you added the water, they called it priming the pump. because when you prime something, that means you start it. So you add a little bit of water to prime the pump, and then it will work and pull out as much water as you darn well please. That's the idea over here with the primer. The primer, its job is to start the strand. So Primase builds the primer. Okay, but here's an interesting thing. The primer in DNA replication here is not made out of DNA. Notice I've got it drawn in pink here, not blue. It's not made out of DNA. It's paired with DNA, but it's not made out of DNA. In fact, it's made out of RNA. And we'll learn more about RNA in week 5. It's pretty much the same thing as DNA. There are a few differences. For now, just remember that the primer in DNA replication is made out of RNA. All right, so the primer in DNA replication is made out of RNA, and God only knows why. The reason the primer is made out of RNA in DNA replication in the cell is simply because that's the only kind of bricks Primase knows how to work with. So we deal with it. So we come in here and we build an RNA primer at a couple of random points along the strand. And these primers range in length from like 10 to 20 or 10 to 30. Some sources say 15 to 30, somewhere in that range, like the 10 to 30 base pairs long. But yeah, they vary. Sometimes it says 15 to 30, sometimes it says 10 to 20, but it's something in about that range of 10 to 30 base pairs long, the primer there. Okay. Sorry, we beat primers to death there. So now that we've got the primer on there, we now have a little bit of a strand that DNA polymerase can come and hop on, and or DNA polymerase III can now make longer. So now DNA polymerase III can hop on and make this thing longer. And I'm going to draw from here on out just to help make a point here. So DNA polymerase III is going to come and hop on at this primer here. And this primer right here. So these are my DNA polymerase IIIs, or sometimes we abbreviate it DNA pol 3. And their whole job is they're going to hop on at the primer, and then they're going to build the new DNA strand. But these DNA polymerase IIIs, they have another caveat. They only know how to build in the 5' to 3' direction. Now, if you remember from week 2, we learned that each DNA strand has a 5' end and a 3' end, 5' end and a 3' end. These guys only know how to build in the 5' to 3' direction. Now, here's a point to make, because we need to distinguish which strand we're talking about. Because remember, DNA strands are anti-parallel. So if I've got a strand down here that I'm trying to copy, and I've got the 5' end over here and the 3' end over here, the new strand is going to have a 3' end up here and a 5' end up here. So do I build, so in this little circumstance, would I build right to left or left to right? Because we're building 5' to 3'. When we say it builds 5' to 3', we're talking about the direction it is building. So the new strand. So in my little example here, it would start over here and build that way, because we are building in the 5' to 3' direction from the perspective of the strand I am currently constructing. So the way that I remember this, that I teach students how to think about this, is we read 3 to 5. We build 5 to 3. Because notice, if I'm traveling right to left on this picture here, I'm reading the blue strand, the original strand. We call it the template strand. There's a good vocab word to know. The template strand is whichever of the two strands is the original strand, the one that pre-existed. So we're reading the template strand in the 3' to 5' direction, because that's right to left, but we're building the new strand in the 5' to 3' direction. So our DNA polymerase III only knows how to build 5' to 3'. And notice that's not always going to be right to left, and it's not always left to right. Left and right are completely arbitrary. And the new strand isn't going to be always the top one, and it's not always going to be the bottom one. Again, it's all about your perspective. So watch your 5s and 3s, and usually we will tell you what the templates, which one's the template strand, okay? All right. So DNA polymerase III hops on at the primer and builds in the 5' to 3' direction. So this guy, so notice there's the 3' end of this strand over here, and here's the 5' end of this strand over here. So that means that for this new top strand here, this is the 5' end of the primer, and this is the 3' end of the primer over here. So we're going to build 5 to 3. So this polymerase is going to build the new DNA strand going this way. And this polymerase, notice he starts at a primer up here. This is the 3' end of the top strand over here and the 5' end of the top strand over here, which means that he's going to, so that means this is the 5' end of that primer and the 3' end of that primer. So he's going to build this way. Okay. And these guys just build and build and build as long as there's a single strand. So they're just going to follow Helicase off into eternity, right? Until they run into another DNA polymerase III or they fall off the end. That's how these guys work. They're just going to keep building. So they're going to read the template strand. And if the template strand has a T, they're going to add an A to pair with it. If it has a C, they're going to add a G to pair with it. So you read the template strand and you add whatever base pairs with it. And that's how DNA polymerase III is able to build the new strand, because thanks to the base pairing rules, DNA polymerase III always knows what base is supposed to go in the new strand based on its complementary base on the other strand. So now that we've got that, now notice we have a predicament again. I've got a helicase traveling that way. So notice helicase doesn't care about 5' and 3'. He's riding on both strands. So we've got one traveling that way and we've got one traveling that way, right? Well, notice that we can't build in the 3' to 5' direction. So helicase starts unwinding and this guy goes and builds and follows that guy, and this guy goes and builds and follows that way. But notice I've got unzipped strand right here and unzipped strand right here that the polymerase hasn't gotten to because it's before the primer. So helicase has exposed it, it's single stranded, it's ready to be duplicated, but the polymerase, it's upstream, as we call it, it's before the primer, which means DNA polymerase III can't get to it because we can't travel in the 3' to 5' direction. So how the heck do we copy that strand and that strand right there? Well, we have to bring back in Primase. So Primase comes in here and it's going to build a new primer right behind Helicase and a new primer right behind Helicase. And then the polymerase III is going to come and hop on here and build from that primer to the back of that primer. Let me show you. Clear my drawings here. Okay, so here's our DNA polymerase III, and it looks like my simulator is not quite caught up to par here, but then it will also hop on over here and over here. And then we're going to build. So we build. And notice, as we build, we're knocking the single-stranded binding proteins off because now we don't need them anymore because the strand is double-stranded, so it's happy. It doesn't want to go stick to that other guy again. So as we travel along, DNA polymerase III is knocking the single-stranded binding proteins off. Okay, so notice, and this would continue building. So the DNA polymerase III would be traveling right behind the helicase over here and right behind the helicase over here. And it would just keep building those DNA strands. So yeah, just bear with my little simulator here. It's imperfect. But yeah, they would just keep right on building and following those helicases that are traveling off that way. And then notice this, we had this polymerase start at that primer and build to the back of that primer, and this polymerase start at that primer and build to the back of that one. And then the helicases will unzip even further, and we'll build another primer and build to the back of that one, and another primer and build to the back of that one, over and over and over and over and over again until we've completed the whole thing. Now that's the, so now we've duplicated these strands. Now let's pause here for a sec and bring in some new vocab words here. Right here in the middle, this was our origin of replication. If you remember, that's where the helicase is bound. So origin of replication. Now let's look on just this side. So ignore this side for a second. Let's just look to the left of the origin of replication. Notice we've got one strand where the polymerase building 5' to 3' is traveling in the same direction as helicase, right? On this top strand up here, in order to build 5 to 3, because since that's the 3' of the top strand, so that means this is the 5' of the new strand and the 3' of the new strand. So we're going to build this way, and helicase happens to be traveling that way. So on this top strand, the polymerase only needs one primer. It gets one primer and it's just going to build for eternity because it's traveling in the same direction as helicase. So we never need to hop off and start over again. So it just builds on forever and ever and ever. We call that strand the strand where you only need one primer because the polymerase is following the helicase. It's traveling in the same direction. So you build it in one long continuous segment. We call that the leading strand. And that's the one where the polymerase can only use one primer and it can build it in one big long segment because it's going in the same direction as helicase is traveling. So it can just follow the helicase to the end of time. The other strand does not have that luxury because 5 to 3, notice there's the 3' end of the bottom one. So we've got, there's the 5' end there and the 3' there. Notice in relation to this helicase right here, because remember, we're only focusing on the left side of the origin of replication here. According to this helicase, this polymerase is traveling in the wrong direction. He's going the opposite direction of the helicase. So we have to build this bottom strand, or we have to build this strand. In this case, it's the bottom one. It's not always. Remember, bottom and top are arbitrary. It's whichever strand the 5' and or traveling 5', building 5' to 3' is going in the opposite direction of helicase. So this strand is traveling in the opposite direction of helicase. We call that the lagging strand because it's going to take a little bit longer because we have to build a lot of primers and build this thing in chunks as the strand gets exposed. So we will start right behind the helicase and travel to the back of the primer. And then in the process of doing that, helicase has exposed more strand that we haven't gotten to yet. So we'll go back and we'll build a new primer and yada, yada, and we'll do that over and over and over again. So that's the lagging strand, the one where polymerase has to travel in the opposite direction of helicase. So the leading strand in this case was the top one, but it's not always. But now notice, so yeah, we've got so the top one in this case is the leading strand. The bottom one in this case is the lagging strand. But now notice, on the opposite side of the origin of replication, so now let's go look at the right side, helicase is traveling in the opposite direction. So now it's this bottom strand that's the lucky guy because we only need one primer because this is the 5' end of the new strand and here's the 3' end of the new strand. So we're now building this way, which just happens to be in the same direction as helicase. So now this is the leading strand. And up here, this one's now the one that's building in the wrong direction. So this is now the lagging strand. So depending on which side of the origin of replication you're on, both strands at some point are the leading strand and the lagging strand. It just depends on which helicase you're following. And that's a concept that confuses, or that a lot of students get confused when we oversimplify this and just focus on one thing, because we say, okay, one of the strands is the leading and one of the strands is the lagging. And they're like, but wait, what about the other side? So that's why I like to show it this way, is in fact, both strands, depending on which side of the origin of replication you are, both strands take a turn being the lagging and the leading strand. But in relation to one fork, one of the two strands is the leading strand and the other strand is the lagging strand. Okay, and the lagging strand is the one that you have to build in segments because it's traveling backwards according to Helicase. And the leading strand is the one that you can build in one continuous piece because polymerase is following the Helicase. Okay, now we can get back to our replication story. So now we've got another predicament. So yes, we've built this thing. We've got two completely separate DNA strands. These things will completely separate from each other and we will wind up with a DNA double helix at the top and a DNA double helix at the bottom. There's your duplicated chromosomes. We can go through meiosis and everyone's happy. But we've got the issue of our primers here. They're not made out of DNA. Remember, Primase only knew how to build out of RNA. So we've got these RNA primers here. So we've got an RNA DNA hybrid going on here, and that's bad because I want double-stranded DNA. I want my whole chromosome to be built out of DNA. DNA is an extremely stable molecule. We'll learn next week that you can pull DNA out of mummies that's still intact. You can pull DNA out of dinosaurs sometimes that's mostly intact, or partially intact at least. So DNA double strands, or double-stranded helixes, sorry, my brain's not working today. Double-stranded DNA helixes can last for like ever, but RNA lasts about 5 minutes. It's not very stable. So this RNA will break down and fall away, and I'll have a little hole in my strand if I'm not careful, and that's way bad. That's way bad. We don't want holes in my double-stranded DNA helix. That's way no good. So we need to replace that primer with DNA. But we have to get rid of the primer first. So we have to bring in another enzyme called RNAse H. Isn't that tricky? RNAse H. So it's pronounced with 2 A's, but there's only ever 1 A. And sometimes they capitalize the A to make it part of the RNA. And sometimes they lowercase the A to make it part of the ACE. But it's pronounced RNAse H. And RNAse H, his whole job is to bind on and cut out the RNA primer. That's his whole job. So he hops on every time we have an RNA primer, and he comes in and cuts them out. So now we've got these little gaps in our new strand where the primers used to be. So now we have to fill in the gaps. And the guy that does that is DNA Polymerase III, the guy that built the original strand. It's DNA Polymerase III. But you remember, but you might be asking, well, how does DNA Polymerase III work if it doesn't have a primer? Because the whole goal is we just cut out the primers. So how is it going to work? Well, the whole point of the primer was DNA Polymerase III had to have a previously existing strand it could make longer. Well, look, I have a previously existing strand now. So this new strand here basically pretends as my primer. So DNA Polymerase III can now hop on here and build out the, well, I guess it would be this one over here because we would have to build in the 5' to 3' direction. So it would hop on over here. So the Polymerase III would hop on over here and it will come in and build from here to there to fill in that gap with DNA. Notice this is now DNA. And to fill in that gap right there. And we'll come up here and fill in this gap right here. And this gap right here. And ta-da, we have a perfect double-stranded DNA helix, 2 of them. So we had RNAse H come in and cut out my RNA primers because we don't need them anymore. And then we come in and use DNA Polymerase III to come in and fill in the gaps that were left behind by the primers with DNA. So now we've got a nice, stable, complete double-stranded DNA helix, 2 of them, and there's my duplicated chromosomes. Now we've got one more step to this, and this is the other one that's usually confusing to people because the books don't cover it very easily. So let me give you a bit of the background story here. This last enzyme, actually, let me introduce the idea here first. Let's come back here. Okay. Last week, we taught you the structure of a DNA nucleotide. So we've got a little sugar here. And then you've got your little base, and it's either got 1 ring or 2 rings. So there's your base, your sugar, and your phosphate poking off out here. All right? You're probably so sick of seeing that. Well, it turns out that this story here is only partially true. Don't you love it when we do that to you? It's going to happen a lot more. Welcome to the sciences. This is the structure of a DNA base that is actually in a strand already. But when it's just a brick that hasn't been incorporated into a DNA double strand, it doesn't exactly look like this. It actually has 3 phosphates right in a row. Isn't that bizarre? So when it's actually built into the strand, it's only got the 1 phosphate, and we link phosphate, sugar, etc. When we do, but when it's just the bricks floating around, they actually have 3 phosphates. And this, you don't really need to know this. I'm just giving you some background here so that you understand this next enzyme better. The reason why you need these 2 extra phosphates is they pack a lot of energy. This is basically a battery pack. So in order to make any chemical reaction happen, you have to have energy. These 2 phosphates here pack energy. So this is essentially a battery pack that can be used to hook this base to the next one. So what polymerase does is, here, let me come in here and erase my little labels here. So we've got, here's the next base in the chain. We've got the chain coming up here. We got phosphate connected to sugar, connected to phosphate. Then we got the little bases coming off the middle here. Okay, there we go. So here's our DNA strand that I'm DNA polymerase now, and I'm trying to add this base to the 3' end, because remember, we start at the 5' end up here, and we're building in the 5' to 3' direction, which means I always have to add the next base to the 3' end of the strand. So we've got to somehow make a connection between this phosphate and that sugar. And these guys have the energy to do it. So DNA polymerase comes in here and grabs this new nucleotide that's supposed to be added on, and it chops off these 2 phosphates, and all of that energy that we just released gets funneled into making this bond right here. And now we have connected the next base on, and we would do the same thing down here. So we would chop off the 2 phosphates to add the next base on, and the next base and the next base, etc. Now here's the point. When I come in here and I cut out the primer, I wind up with this predicament here. So let's pretend that this base represents the primer, right? Remember, the primer is actually 10 bases long, but just to show a point here, let's come in and pretend that this base was the primer. So RNAse H comes in here and cuts it out. Okay, so now we've got this gap here in the strand. Okay, use your imagination. It's more than 1 base. All right, we got this gap strand. This one's DNA, that one's DNA. Now polymerase has to come in here and fill it in again. So now we bring in my little base here to come in and fill in the gap. Okay, so he's got his 3 phosphates so that we can add him in. So this is the new base. So we cut off these 2 phosphates right here, and we glue him to that one. That's great. But do you see our problem here? How do I hook that one to that one? Where's the energy going to come from? We spent this guy's battery hooking him to the primer that we just cut out. He doesn't have a battery. So I can't hook this phosphate to that sugar without a battery, but we spent his battery already. So somehow I need, because I can't have a nick in the backbone here. That's not okay. So somehow I need to connect this phosphate to that sugar, and polymerase can't do it. So we have to introduce our new enzyme that comes in here. And his name is Ligase. Or sometimes he is called DNA ligase. They're the same thing. Ligase is essentially a glue gun. So he brings in an external battery pack. He's got the jumper cables. So he comes in and his whole job is to make that bond right there. Ding. So he just grabs that phosphate and that sugar and sticks them together. So notice he's not adding any new bases. That's a common misconception. Ligase doesn't add any bases. His whole job is after we make that, after we cut out the primer and we fill it in again, his whole job is to just link those last 2 bases that polymerase can't do. That's his whole job in life. They usually say his job is to fix the nicks in the backbone. But that's what he's doing, is he's coming in here and providing the energy to link those 2 bases together. But polymerase put the bases in, but those last 2 bases he can't hook together, so ligase has to do it. So yeah, that's the last guy in the line is DNA ligase. And then you're done. Now we're done. Isn't that awesome? Okay, one other vocab thing that I failed to cover back here. So let's come back here and cover. Let me build back to the point that I want. Okay. Okay, here we go. This is where I want to be. All right. Notice on the lagging strand, so we talked about how you have to build the lagging strand in chunks, right? We've got, we have this piece right here. And then we would have another piece and another piece, right? We have to build this thing in pieces. So the leading strand, we just build in one chunk. This one we have to build in several chunks. These little pieces are called Okazaki fragments. An Okazaki fragment is simply the segment of DNA between 2 primers. And they are only found on the lagging strand. Because remember, the leading strand is built in one big piece. So we don't have these little between 2 primers things going on because we build it all in one piece. There's only one primer on the leading strand. So Okazaki fragments are only on the lagging strand. And the definition is they're the segment of DNA between two primers. So it's the pieces that we build. So the whole job of, so we cut out then the primers and we'll link the Okazaki fragments together and poof, now we're all done. So yeah, that's DNA replication in the cell. That's pretty intense, huh? So yeah, let's do a quick recap of all the enzymes that we expect you to know. We expect you to know DNA Polymerase III. That one makes the DNA, or that one is responsible for building the new strand. Make my pen a little skinnier here. Okay. Then we've got Helicase, which was responsible for unwinding the strand. Unwinding, I can spell. The helix. And then we had let's see here, we had Topoisomerase, which was to fix the kinks or remove the kinks ahead of Helicase to make sure it didn't get stuck. And then we had Primase. Oh, sorry, we had single-stranded binding protein. Let's hit that one first. We had the single-stranded binding protein, which kept the separated strands apart. And then we had Primase, which built the RNA primer. And then we had, so DNA Polymerase III came in and built the new strand. And then we had RNAse H, which removed RNA primer. And then we've got Ligase that fixed the nicks in the backbone. Backbone. Those are the enzymes of DNA replication. Isn't that great? One other point before we finish. Sorry, this video has ended up being extremely long. After you've duplicated DNA, we have to make sure that it stays good and that DNA Polymerase III didn't make any mistakes, because he does make mistakes. About 1 in every 3,000 base pairs or so, 3 million base pairs. I can't remember whether it's 3 million or 3,000, but he occasionally makes a mistake, meaning he puts in the wrong base. So like he reads an A and he accidentally puts in a G instead of a T like he's supposed to. So we need somebody going through and checking for those and editing the DNA to make sure that he does, or that we don't get any mistakes, because we'll learn in week 5 that even a 1 base typo in the DNA strand can really mess things up. So we need to make sure that the DNA Polymerase III did his job right. So we have another enzyme called DNA Polymerase I. Isn't that awful? Not to be confused with DNA Polymerase III. Okay, we've got, I'll even put them in a different color here to make sure. DNA Polymerase I. One is the DNA repair / editing enzyme. /editing. So he goes through and makes sure we spelled everything right. And if we spelled something wrong, if we have an A paired with a G, he will cut out the bad base and put in a good base or put in the base that's supposed to be there. And that's it. That's all of them. So go watch my other video to figure out how PCR works. And yeah, thanks for sticking through to the end. And hopefully this helps. If you have any questions, feel free to reach out to me. My email is dallin.stokes17@gmail.com. And yeah, you can email me, or feel free to set up a tutoring appointment or reach out to your professor. We're happy to help. So thanks for watching.